Okay, now that we have learned about osmosis and water potential, we're going to apply that to talk specifically about what happens when water moves across a plant cell membrane. Water potential affects the uptake and loss of water by plant cells. So some plant cells will have high water potential, in which case they'll be likely to lose water. And other cells will have low water potential, and they'll be more likely to uptake water. So you can envision that those two different types of plant cells might be found in opposite or different parts of the plant, and we will talk about sort of how that works in one of the later lectures. But first we want to talk just specifically about what happens to a plant cell that has either relatively low water potential or relatively high water potential. So there's several different scenarios we want to talk about. First, we're going to talk about the scenario where we have a flaccid or limp plant cell. And all that means is that the central vacuole that usually has a lot of water in it doesn't have a lot of water in it. And a cell like that typically has low water potential, meaning that water is going to want to move into it. However, when you place a plant cell like that into an environment with an even lower water potential, so an environment that maybe has a whole lot of solutes in it, that cell, which is already flaccid, is going to lose more water, even though it doesn't have very much, and it's going to undergo plasmolysis. Plasmolysis is the process by which the cell loses water and the cell membrane actually shrinks away from the cell wall. I'm pretty sure you guys saw that when in lab when you were looking at the Elodia cells, but I'm going to show you a video of that again in case you don't remember or in case you didn't get to it. So you just have to wait a second. The other scenario is if you have a flaccid cell. Again, it's a cell that doesn't have a lot of water in its central vacuole, and you place that into an environment that has a higher water potential or low solutes in it. In that case, the cell has the lower water potential, and water is going to move into the cell, and it's going to gain water, and then the cell becomes turgid. And what that means is the central vacuole fills up with water, and the cell membrane sort of swells and pushes against the cell wall so that the cell becomes really firm. So we are going to see some diagrams and some videos of that. So this is from that first example where we had the flaccid cell. And again, the flaccid cell actually has low water potential, where you would expect water would want to move into this cell. However, if we put it in an environment that has an even lower water potential, then you end up with the initial conditions of the cellular water potential is higher than the environmental water potential. So let's just say we put a flaccid cell into an environment that has really high levels of solutes in it. If that happens, then water from inside the cell, which has a higher water potential, is going to move out into the environment until the water potential on both sides of the membrane are equal. When that happens, again, the cell membrane pulls away from the cell wall, and that is called plasmolysis. So let's watch this video. I can't get that video. Oh, there it goes. Okay. So this is what you probably saw in lab. You can see the cell walls shrinking, cell membrane shrinking away from the cell walls as you put the cell or the cells into an environment that has a lot of solutes. So an environment with a lower water potential. So the video goes on a little bit longer, but you're not going to see a whole lot more action from that. You are able to replay any of these videos, so if you didn't get to see that, you can come back and look at that again. Okay, so now let's look at the second scenario that we talked about. In this case, you still have this cell that doesn't have a lot of water in its central vacuole. And in this case, you're going to place it into a solution that has a lower solute concentration than the cell, which means that this cell will have a lower water potential than the environment that it's placed in. So here you go, you've got the cell, the low water potential, high water potential in the environment. In this case, this is pure water with no solutes in it. And remember that areas with high water potential, in that case, the water is going to flow from the high water potential to the low water potential. So in this case, water is going to flow into the cell and cause it to become turgid. So let's watch this video. Okay, in this case, you can see, well, you can't really tell, but these are the central vacuoles, which are filling up with water, and they're squeezing all these little organelles. These chloroplasts are sort of becoming squished together as the central vacuole gets bigger, and it's pushing the cell membrane against the cell wall. So let's watch that for a few more minutes. Okay, so you've got cells undergoing plasmolysis in the last slide, and in this case, you have cells that are becoming turgid with water rushing in if the cell has a lower water potential than its surroundings. And this is important because this tendency of water to flow into and out of a plant cell has a lot to do with how transport happens from the roots to the leaves through the vascular tissue, specifically the xylem.